in Paris, as you heard, so it's quite attractive. So welcome. And my name is Emily, and we are in the team. You already heard Petra. She's online. She's supporting us, and Tammy also. Here, yes. And you are in the session of CoBioTech, the call. So we normally would have 4 projects here, then it's decreased to 3, and now I heard that the middle one also is not there. So we now in a very comfortable situation that we have a lot of time. I was already asked whether we could have a bit longer presentations. So I would think that it's good to have both presentation right behind each other, and then have a question round for the 2 projects. And yes, we will have to record we will record these presentations, and then afterwards you can have a look at them. It will be available. Okay, I think. Yeah, after this presentation, I have to say first lunch. So that's not so interesting for the people following us online, but for the people here. So at 12:15, we will have lunch, and then meet again in Plenum 1 or Berlin 1 at 1:15. Okay, I would say just start. We'll change this. We start with 3 plus. That's the same order as we had already. Thank you very much, and the floor is yours. Thank you.
Thanks a lot, and thanks for doing this session today. I imagine everything's working well at home, and you can hear me well, and the slides are sharing, and all of that. If not, give us a shout. Actually, someone monitoring. All right, so I'm going to give an overview of one of the projects that run as part of the second call. It's called My Place, and that stands for Microbial Integration of Plastics in the Circular Economy. As I said in the pitch, we've been working with plastic waste. I'm the project coordinator, and the project involves partners in 5 partners, 4 different countries, 2 in Germany, the Universities of Aachen and Leipzig. We had 1 partner in Spain, and then there was a company involved as well in France, and they specialize in making polyurethane. So the project finished about a year and a half ago. It was a bit impacted by the pandemic at the beginning, but we managed to catch up, and I want to give a few highlights. But before getting into that, just to give a bit of an overview of the problematic we have with plastic, with this graph, I just want to illustrate that in the last 20 years ago, we've made more than half of the plastics that have ever been made. And plastics as a material, they have been commercialized since the late 1940s. And sadly, we are making more and more and more every year. I mean, despite our efforts to try to recycle, reduce, reuse, etc., that will tell our children, I have little children, they tell them that's cool. We are still making more because they are missing materials, and we make them for lots of things. They are great, for example, for things like the building environment. So we use them a lot for insulation. It's not only the plastic packaging that we always have in mind, there's a single use and it's a problem for the environment. The thing is that the problem with them, so they're amazing, but they last forever, up to the point in which about 5 years ago, there was an issue with National Geographic. There was this talk about how the planet is turning into a plastic planet and how there are going to be more plastic pieces of plastic in the ocean than fish by 2050, which looks like it's a bit of an overestimate, but it's not far off. What you may not remember is that this issue of National Geographic was beautifully wrapped in 3 layers of polyethylene itself, which kind of defeats the purpose of raising awareness against the problem. Okay, so this is just to give an overview of what the project was about. So we wanted to take two plastic types out of the many that are commonly used. Polyesters and polyamides, I mean, they're very difficult to deal with, but they are the easiest, just because of the presence of these heteroatoms that we have in the structure of the polymer. We do work with polyolefins in my group right now, and we're working with companies trying to make biodegradable polyethylene. But for my place, we focused on polyesters, which is what most people are doing these days anyway. And the idea was to try to leverage enzymatic activities that we know exist and break down the plastics, and they make the constituent monomers. So in particular, for PET, for polyethylene terephthalate, we can express these PET hydrolases and then recover the constituent monomers, terephthalic acid and ethylene glycol, that they can be combined together, and there are industrial processes for this. You may have heard of a company, French company called CoBioTech2. They do this enzymatic industrial processing of PET pellets. But in our project, we were interested in actually upcycling these monomers and try to make some of the molecules. And we had a set of targets, things like adipic acid, different types of butadienyls, hydroxyalkanoic acids. What they all share in common is that they are bifunctional. And this is important because it means that we can combine them and then try to put them together and create new materials. And in particular, we wanted to create polyurethanes that incorporated these bio elements, these building blocks that come from renewable sources. So that's kind of the overview. Just to show a few of the highlights of the results that we got, we went into the environment and we screened for organisms that could break down PET more efficiently. We couldn't isolate microorganisms that themselves were able to grow using the plastic as sole carbon source, but we found specific metagenomes, really interesting enzymes, and in particular one of them that is called DHL7, you have in here, is able to outperform under the conditions in which it was tested the gold standard for the enzymatic recycling industry, that is called LCC. As it usually happens, the devil is in the details, and it turns out that DHL7 for its activity needs lots of phosphate. That is not something that is present in the industrial process itself, but if we were thinking of using these enzymes for breaking down plastics that then could be straight away fed for microbes, then it would be a great thing to have. In terms of what to make out of the plastic, so I'm not sure if I can. There are different applications that we targeted. This is just one of the examples, and it shows the production of adipic acid. So we took an organism that was engineered to feed on the terephthalate that comes from PET breakdown. And then we took advantage of some of the enzymatic reactions that are already present in this bacterium, and we added a few extra. We removed some of the competing pathways, and eventually we got to the production of adipic acid. This process could be scaled up to an extent. So we ended up using a close to 5,000 liter bioreactor in order to produce a few grams of adipic acid that come from plastic. It's not impressive, but it's a start. And this adipic acid can actually be incorporated into polyurethanes. Despite our efforts to scale this up further, this is as far as we could go during the duration of the project. In addition to adipic acid, we made other things like biolacin, I'll show an example later, bioplastics like polyhydroxyalkanoids and hydroxyalkanoic acid. So there were a few examples of upcycling of terephthalate and ethylene glycol. Now, another important element, that this was the commercial interest of the company involved, is whether these new building blocks that we can get from renewable sources can be used for making materials that are actually used. And some of these were incorporated into polyurethanes. And then the resulting, so we characterized the resulting materials to see what were their mechanical properties. And I have a few examples here. So ethylene glycol is already commercially used for making standard polyurethane. So you can think of that one as a benchmark. And what I'm showing here is a stress response test. So the higher this value, the better the material, let's say. Out of the butadienyls that we thought of making, it turns out that some of the isomers are not great, because we get very brittle materials, materials that are not really mechanically very good. And only the mesoform is something that could match in terms of performance what can be achieved by some of the conventional monomers that are used for polyurethane. But this was really unraveling. Okay, we did a lot of work on responsible research and innovation. And this helped us to kind of drive the direction of the project towards things that were probably more useful than what we had initially thought. So we tend to think of plastics as something very big pieces of plastics that accumulate in the environment and they are detrimental for the fauna, the flora, humans, etc. But we realized that the real danger is in the microplastics. And these microplastics can emerge from the decomposition of the large plastics. But to be honest, one of the main sources of microplastic accumulation in the environment are the microplastics that that accumulate that come from textiles. So these are pieces of fabrics, fibers, etc. If you think of our clothes, most of them are made with polyesters. These polyesters, some of them are synthetic, including ethylene terephthalate, etc. And we identified that at least in the United Kingdom, we have a problem with microplastics that are accumulated as part of the wastewater treatment because they end up being part of the sludge. This sludge, after the wastewater treatment process, is used as a fertilizer. So we just drop in fields. And therefore, that's a way in which microplastics are released into the environment. It's not because they are miscollected, it's not because they go to landfill, it's because they wash off our clothes. So we thought that this could be a problem that we need to tackle. I don't think that you have this problem in Germany because everything gets incinerated at some point, and the sludge that is recovered is not just a fertilizer, but in some other countries, we use 80% of our natural fertilizer comes from wastewater treatment plant in the UK. So it would be great to have microorganisms, so engineered microorganisms that can feed on these microplastics directly. So that was something that we tried to do in the project as well. And what we did was to take some of the organisms that we could get from the environment that are able to at least grow on terephthalic acid as the sole carbon source, and we engineered them to express different enzymes. And I'm not sure if you follow this field very often, but whenever, every now and then there is an enzyme that appears in the news that is much better and faster than the last, and it's been maybe engineered by artificial intelligence or different methods. And just to be told, is we don't really have a very good standard assessment of how good they are. And it all depends really on the conditions. So if we were to engineer micros that can make these enzymes, we need to test all these enzymes in the same conditions and be able to run a side-by-side comparison of all of them. So that's something that you have on the screen right now. We took a couple of our favorite organisms. One of them is E. coli that we use all the time for absolutely everything in the lab. The other one is a Pseudomonas strain that we got from the garden on campus and is able to feed on terephthalate. And then this one was modified to secrete one of these enzymes. Different of these enzymes. And you have the colonies at the center of the plate, and those halos surrounding the colonies represent the breakdown of not PET, but an analog that is polycaprolactone. So the greater the halo, the greater the secretion and activity. And I have one of them in red, which is PHL7, that was discovered as part of the project, that under those conditions is the best. So we tried that. And then we tested not any time of polyethylene terephthalate, but a big part of the project was about what are the best pretreatments that we can conduct on the plastic so that it becomes really bioavailable and accessible for enzymatic breakdown. We tried a number of things, and you have some represented here. So we tested films, amorphous films as they can be sold. We cryo-milled them, we sieved them to generate microplastics, and then we took films that were crystalline, that were post-consumer, that we get from the supermarket, and we dissolved them with an organic solvent. And then we tested the activity of all these enzymes that have been previously secreted by a microorganism to see which one is the best. And you have some results here. The higher the bar, the better the activity. It turns out that out of all the plastic types, the one in which we can get the highest performance, regardless of the enzyme that we use, is that solvent-treated plastic. And the same thing is represented here when we check those results by mass spectrometry. Now, the question is whether the solvent treatment is more sustainable and not doing anything. And it's something that we are trying to answer by doing different type of studies like life cycle assessments and techno-economic analysis as well. Okay, so now that we have our favorite organisms and we have our favorite enzymes and we have our favorite pretreatment, we wanted to see whether we can combine all of them and trying to obtain a microorganism that can actually survive using the plastic as a feedstock. And these are the results you have on the slide right now. And you can see that after 4 weeks of incubation, with the best organism that we could come up with, we have something that we call moderate petrophy. So there is at least some biomass that is being generated, even though it's not impressive. So just to conclude, as part of the project, we show that it is true that we can break down plastics enzymatically. We can use these hydrolytic products to feed microbes to make other things of value. But if you are worried about, and this is a question that we get from journalists a lot, if you are worried about whether these microorganisms, if they were to be released in the environment, they are going to lead to the collapse of civilization because they're going to eat all the plastics, I don't think that's going to happen anytime soon. So just to conclude, just wanted to thank everyone that took part in the project, and of course, your attention.
Do we do questions after? Yeah, we do questions after. The fairest way to do it then. Yeah, thanks. 15 minutes. Hello, everyone again. Thank you very much for joining our Paris room. I'll be presenting on the Cyclone project, Synthetic Microbial Communities for Sustainable Production of Food Ingredients. And I want to start by thanking and naming the partners involved. It's been a collaboration of universities and industry partners from Spain, Germany, UK, and also Slovenia. And I'm the representative of the project coordinator, Rodrigo Ledesma Amaro. I recently joined his lab as a research associate, and our lab is specializing on synthetic biology, and this will also be the main focus of me sharing insights from the Cyclone project. So as I mentioned already in my pitch, engineering biology or synthetic biology is this emerging discipline that applies engineering principles to living systems. And really the trick here is to allow us to redesign living systems that can be cells or whole organisms in a way that they produce valuable compounds for us, so for industrial and economic use. And one example for that is the engineering of microbes for industrial production. And so the Cyclone project was aiming to produce the chemical limonene with the help of microbes. And so just a few words on limonene itself. I want to show a lot, so I'm just wondering. Yes. So you got the chemical structure here. It's a cyclic terpene. And you're probably all very much familiar with the smell of limonene, because if you peel an orange, then this is what smells like citrus, limonene. But in industry, it's actually a very versatile compound. It's used in the food industry, pharmaceuticals, and even materials industry itself. And then also you can derive derivatives from it that are used in a variety of cases. So traditionally, limonene is extracted from plants. As I was saying, you can find it in oranges, but this extraction process is not that much efficient. And also it has the disadvantage that it's actually dependent on the harvesting cycle of citrus fruits.
An alternative could be that the limonene can also be produced through chemical synthesis. But also here the downside is that that's quite an energy-intense process. So this sets the stage for the need for alternative ways for the chemical manufacturing of limonene. And this is exactly the goal of the Cycling project, where we want to use microbes to convert low-cost carbon sources to this high-value compound limonene. And so an example for these low-cost carbon sources that are ideally industrial byproducts, very abundant, could be glycerol. And with the help of an engineered microbe, we now want to convert this into this high-value compound. How to go about that? So our host of choice, our microbe of choice, is a yeast called Yerovia lipolytica. And so it's not a conventional yeast, so usually people don't work with it, but it has many advantages. On the one hand, it is very robust, so it grows very easily. It provides a lipid environment, so it's very high in fats, and this is particularly good for terpenes like limonene. Also, it has the regulatory status as safe. So this is our host of choice here that we are going for, Yerovia. But so how do we go about now to produce something that Yerovia usually doesn't produce? So Yerovia endogenously does not make limonene. And let's first look into how plants naturally make limonene in their cells. So the ingredients for limonene production are, on the one hand, you need the direct precursor molecule. In this case, it's a GPP. And you need the enzyme that converts the precursor to the limonene. And that one is called limonene synthase. So now, if we go back to our yeast, now we apply metabolic engineering, turn this microbe into a biofactory for limonene. So luckily it has already the direct precursor molecule, because it's also involved in other metabolic pathways. And we now genetically complement the yeast with the enzyme, the limonene synthase, to then make limonene. But unfortunately, this simple approach is not really enough, because it could work, it does work, but we heard already, it's everything about pushing efficiency. And this is not very efficient, because it turns out that GPP abundant in the cells actually is not very high. So as metabolic engineers, we think about ways how can we upregulate abundance of this to then boost the limonene production. So if we look a little bit into the context of the metabolic system in the yeast, the GPP itself is made through a pathway called the mevalonate pathway from acetyl-CoA. And so as part of the study that was, by the way, recently published from our lab, the authors brought in different enzymes. I'm just naming the names here, it's a bit long, but they basically brought in enzymes that are participating in this pathway and upregulate them. So we had to have a lot of them, that we have lots of conversions from acetyl-CoA to GPP, and then as a result, also more conversion from GPP to limonene. So that's one approach of the metabolic engineering to increase yield. And really all this metabolic approach aims to optimize this conversion from the feedstock, could be glycerol, to our desired end product, whilst not compromising the general fitness and health and well-being of our microbe. Because in reality, it's much more complex. And whenever we tweak a certain metabolic pathway in favor for the production for our desired compound like limonene, this can also have detrimental effect, for example, for growth or general survival of the microbe. So what worked in the end, I'm highlighting here that the authors brought in these four enzymes additionally into the microbe that yielded ultimately an efficient limonene production. And before I show you some numbers on that, I want to name another trick that we applied, because there was another way to further increase limonene production. Looking back now into plants, plants make their limonene not just anywhere, but they have a dedicated compartment called the plastid. And so the advantages of that is if you have all the ingredients for your chemical reaction close by, you actually make it more efficient because they meet each other, and so the conversion is very, very good. And also, in addition, compartmentalization also mitigates the potential cytotoxic effect limonene has if you have a lot of it. So now this logic was applied to the engineered yeast. And the whole pathway that I showed you before was now taken and put into a cellular compartment called the peroxisome. It's generally involved in a variety of metabolic pathways. And so achieving the same effect that now everything is close together, increasing the yield and the efficiency of limonene production. So the resultant best performing strain was then grown in a bioreactor. And just to show you some numbers here, so we see growth over time, and here we measure different things. So for example, over time, the cells grow, so we increase in biomass. With it also the fraction of the limonene that gets produced. And then in terms of the feedstock, so here we don't use glycerol, but glucose as a proof of concept. In the beginning, we have the growth phase, so we have high glucose that the biomass can be generated. And then we switch from growth to production phase, where glucose is held as at low concentration so that the microbes focus less on growing, but actually producing limonene. Okay, and lastly, I'd like to touch on another future perspective of that project, which revolves around the possibility of expanding metabolic capability by co-culturing of microbes. So up until now, we talked about a monoculture, one species, basically an autonomous species. But we know that if we can form a synthetic microbial community, we can actually metabolically expand the capacities that also results again in more efficient production. And just to name an example of that, also a study published recently from our lab, is the production of another chemical, malonic semialdehyde, MSA. And in that study, they used a different yeast. So now we talk about Baker's yeast, Saccharomyces. And so just to go a little bit more into detail of this principle of this co-culturing, because yeast or microbes in general, they like to be independent. So what we as engineers need to do is we need to make them codependent from each other. So in this study, a big library of yeasts were in pairs combined, I think over 3,000 combinations. And these yeasts, they all had a deficiency. So they couldn't really grow on their own, but they needed a certain metabolite in their media to grow. And they did find, I think in less than 3% of the pairs, that the combination actually formed a spontaneous community because they somehow traded whatever they needed with each other. So, but that was quite a rare event. And now in the second step, the pathway to make the MSA was not put in one microbe, like with the limonene, but actually split between the two species of microbes. And it turns out that this splitting of the burden actually increases the yield. So if we look now into how much MSA is made, the wild type, which is the monoculture, performs not so great, but different co-cultures that were mixed in different ratios, so there are all kinds of parameters that play into that as well, they perform much better in terms of MSA production. And so far, the co-culturing is really an underexplored aspect of microbial production. Okay, so these studies that I've shared with you, they were all conducted in an emerging hub for sustainable food production. So I'm part of Imperial College London, and we have 3 new centers starting right now. One is the UKRI Microbial Food Mission Hub. We have the new Bezos Center for Sustainable Food Production and the National Alternative Protein Innovation Center.
Bezos, like Jeff Bezos?
Yes. And so I want to thank you for your attention for now. And my call for action is that we are currently establishing the center. So we are looking for collaborators in a variety of aspects. So we've talked a lot about microbial food, which falls into fermentation. but we also do research on plant-based and cultivated meat products. So please get in touch, speak to me now, you can visit the lab website, and happy to take questions. Thank you.
Thank you very much. Both presentations were really great and inspiring. And now we have the chance, or you have the chance to ask questions. And Greta, are you there? No,
Yes, I'm there.
Sure. I first will have a look here in the room about the questions and then come back to you whether there are questions in the chat. We have quite some time now, as I said, comfortable, and yeah, then I would say just start the question and just say which presentation. Yes.
Okay, so thanks to both. I can start with the first. You said that you do not foresee plastic eating bacteria soon or ever, or how you concluded it. I didn't catch exactly. Is it because it's not efficient, or is it because you think it will never be sustainable, even if it is efficient?
I don't think the degradation that we can get using whole organisms at the moment is fast enough. And there are several limitations for that, but if you think about, for example, how long does it take for a banana peel to completely decompose in the environment, it's not something that happens overnight. So some of those polymers that microorganisms are able to feed on are very difficult to break down. So they managed to do it, and I suspect with plastics something like that could happen. But even the most bioavailable of our plastics, that is this polyester that has been severely treated to make it extremely accessible for the enzymes, is not a great substrate for growth. So I'm not too worried about microorganisms being able to break down the plastic that is already in the environment. Having said that, it is true that enzymatic treatments, for example, are faster just because we can have more of the enzyme if we need to.
Yes.
Lisa?
Yeah, maybe my question is also towards that. So one strategy is to have a microbe and let it secrete the enzyme. And that works, or is it easier to just chemically make the enzyme and then put it on
Okay, define easier.
there? Feasible, I mean,
Yeah, so
many parameters.
the industrial process of enzymatic breakdown right now is we express the enzymes, we make the enzymes in a bioreactor using bacteria, we break the bacteria, we extract the enzymes, we purify them, and then we use them for the hydrolysis. And obviously the advantage of it is that for the hydrolytic process, you can add as much enzyme as you need, and therefore that can speed up the process. Having said that, it's never as fast as the chemical process, by the way. But anyway, I mean, you could think of making that better using more efficient enzymes, having more of the enzyme. But also that comes with a cost. That is that you need to fill your reactor with the microbe that is fed with glucose to make those enzymes. Which in some cases, some LCAs show that that's not very sustainable because you need high temperatures, you need the glucose, you need to purify the enzymes, etc. So we thought one approach could be to just have the microbes that could grow on the plastics themselves, making the enzymes at the same time. And that should make things more sustainable. But when doing, especially talking to lots of stakeholders, the people in the recycling industry, they're very skeptical in general about any bio approach used on plastics. They made us realize that there is a lot of plastics that are actually not even collected. That they end up in the environment. The microplastics that come from textiles and different sources as well. For those, I mean, it doesn't really matter whether you can produce the enzymes or not, because unless you can contain the plastic somewhere that could be treated, there is no other way around them. So we've been advocating for a while to try to think of these technologies and use them in a context that is slightly different. For example, as part of the wastewater treatment plant. We do microbial aerobic digestion of our waste. We do anaerobic digestion at the end of the process as well. Why couldn't we think of having microorganisms that are very good at dealing with the plastics that are present there? So that's kind of how I see this moving forward. The enzymatic hydrolysis, I think, is fantastic. It works with some of the polymers, but not with all. Chemical processes can work as of today at a much higher scale and faster rates as well.
So you're thinking the approach of using microbes to degrade plastics versus degrading it in a different way and then using microbes to convert it into something useful. That the second one is more close right now.
The second part is something similar to what you've tried to do. So once you have whatever comes out of the plastic, then you just need to feed that to the right microorganism, and eventually they will incorporate in the biomass. And obviously when that happens, by using the smart pathways or the right tricks, you can try to boost production yields out of those substrates. So turning terephthalate into other things of value has been done by many people. There are many examples out there. Ethylene glycol is not such a good substrate. We have problems with other types of plastics like polyethylene because the polyolefins, polyethylene, and polypropylene, when they break down, we don't even know what they make. It's a very complex soup of hydrocarbons because there are no building blocks that can be chopped into the constituent monomers anymore. It's just carbon, carbon, carbon as far as you can see. Therefore, you don't even know what's the substrate that is going to get into the microorganism. So it's a different engineering problem. I'm not saying it's not doable. It just requires a different mindset.
I would say just ask the ones online that we integrate them, and then next. Petra?
I think I've got a question from Tim Buck, but for my place. But I think I will now unmute everybody. So Tim, you should be able to pose your question now yourself.
Hello.
Sorry,
I'm not there.
Technique is now working.
I was wondering which you described the engineering to make adipic acid. I wondered which microbe that was and whether it was able to grow on PET alone or whether you need to add glucose.
Hi, Pete. Good to hear from you. It was with Pseudomonas putida KT2440, which I think is one of the ones you've used in the past. And if I remember correctly, the carbon source was terephthalic acid and nothing else.
Wow.
The terephthalate comes from the breakdown of plastic using enzymes in a separate process.
Okay, because I was just thinking if you engineer the flux to make adipic acid, then presumably you have to block some competing pathway, and whether that affects the growth. Yes.
Yeah. So that was done by our.
Still here? We need to switch it on again.
So we can still hear you, but the only thing that is interrupted now is the picture from the room. Don't just continue.
No.
We don't necessarily need to see you.
Isma, do you hear us? I'll go get
We can hear you.
Because perhaps I have to say this technology is working. I love
The only thing we are missing is the picture from the room. So we have the shared screen.
No, it's a TV. It's set with the switch off.
Using the speakers.
Ich habe nichts gemacht. Ja, hallo, Feli, was machst du hier? Doch nicht an. Ah, okay, dann drauf. Ja. Ich habe nichts gemacht. Wir haben da drauf gedrückt. Keiner? Nee, hab ich ausgemacht. Doch. Okay. Oh. Ja. Okay. Immer gut. Tim, are you still there? Do you hear us?
So I'm still there. Tim has muted himself. He's there. I can see he's moving everything. The only thing that we are missing is the picture of you, Emily. We have the shared screen,
Yeah.
So I think just go ahead.
So I think we did pretty well.
Now we have your picture again. Just go ahead. That's really not a point.
Nee, das ist gut so. Okay. Ja. Thank you. I'm not sure. Tim had two questions. They are answered. So then, yeah, I have a comment to the question. My comment is that I don't want to say former synthetic biologist because you never really let it go, but as a micro person, I was really excited about both of your presentations. And also, I think Nina's presentation this morning, I just had so many ideas of things that didn't work for me, and also all the ways you could you could kind of integrate what she's talking about, not working in just one team, and policy aspect when you said, you know, recycling people don't really, and communication and explaining how things work and why they're important. I thought that was a really interesting link for this presentation this morning. I had a question for you about the policy brief paper, and could you just speak a little bit more on that and how it was received and how it came about?
To give a bit of background, we, as part of My Place, we conducted interviews with different stakeholders that kind of focus on different aspects of the plastic producing and recycling, different supply chains as well. And this included consumers, the general public, etc. And as I said, because of the problem is so big, it's really difficult to convince someone that works in the recycling industry that these technologies could work at a scale. So we thought, what could be the best use of our technology? And then when talking to especially water companies that also are responsible in the UK for doing the wastewater treatment that happens after they get to our houses, right? They see microplastics as one of these emergent pollutants that are absolutely everywhere. It's not the only one they have, but microplastics are very prominent. Now, out of all the different plastic types, polyolefins are most abundant than anything else, so 10% also are polyesters, what they think. And they see this as a problem as well because these microplastics get released in the environment. There is no way of getting rid of them as part of the process of making these fertilizers. So we thought, okay, this is an area in which we could have an impact because it's a very defined stream that's leaking into the environment, and we could try to do something and develop technology to deal with it. So we looked into regulatory aspects that apply to microplastics, and it turns out that there is not a lot there. So everyone is kind of expecting that there's going to be very strong regulation limiting the concentrations of microplastics in the environment, but so far it's not regulated. We don't even know if there's even a safe level of microplastics that is something that you could say, you know, we're not worried about having too much or too little. And then we looked as well into what parts of our current treatment processes we could use to have an impact and leverage some of the things that we were doing. And we thought, or maybe as part of the aerobic and anaerobic fermentation processes, we could try using microorganisms to break these plastics down. In terms of reception, it was overall very well received. We haven't influenced any policies as of yet. So no one has reached out from government saying we are going to change regulations, but it's been very useful in terms of creating awareness, and we've done lots of interviews. So lots of people have come asking for, you know, not only journalists that work on science, but also for broader audiences as well. How do you think this is affecting us? Something that came up very often as well is the presence of additives, of which we haven't said anything today, but they also have very detrimental effects for health. Every plastic is made of 1,000 other different plastic types and chemicals. And that was something that we kind of touched upon as well.
So a question to the second project from the audience. Was we talked about plastic a lot.
Yeah,
but,
I have a question actually. So how do you make sure that you have to, industry, I mean, we've been talking about using microbial consortia for absolutely everything for a very long time. And I see chemical companies, but the companies that do bioprocesses, I see them very reluctant to use more than one species if they can avoid it. And one of the reasons for that is because they cannot really. So it's very difficult to control one and to deal with one. You know, when you have more than one, it's almost impossible. So how do you see that, or how are you approaching that?
Yeah, I mean, in the sense that I also get is that they are very difficult to get. Because so in that screen where they screened 3,000 strains, they found a handful that actually formed a community. And this was a pair. I think as soon as we go higher, it's getting even more complicated. And I think recently we also published a study where we were trying to do a computational model that would help to find a pair. And we're only talking about pairs really. And so, but there are so many different parameters of exchange, of growth, that it's very difficult to predict, to be honest. I think that was the take-home message on that. And so I think what was also shown in that study is even though, so one parameter that you could change is the ratio of incubation, but they showed that they tried different ratios, but then it does stabilize at some kind of ratio. So in that terms, maybe there is some kind of flexibility. But yeah, I guess in terms of controlling them, yeah, I can understand that downstream there are complications to just that we need to understand, I believe. But I do think that there's a big advantage in trying, because yeah, so this was now an example where a chemical compound was made with a consortium, but then so I'm joining the food team in our lab, and so there is a lot the question revolving around, can we have communities that make different things that then serve as micro or macronutrients in the food product in the end, instead of mixing them later in the process. So yeah, there are many different applications of this new concept, but we simply don't understand really why do they form a spontaneous community now and not the others. So I think yeah, there's also a fundamental research side that we need to understand a bit more. Yes.
Yes, very specific question. You work with this Jarovia,
Yeah.
yeast. Why did you choose that yeast to produce this terpene? What is usually you would like to have some inherited advantages with respect to what you're aiming to produce. So what could it be here?
Yeah, I mean, so for that, I would say it's the microenvironment, that it has this lipid-rich environment that is favored by terpene. And I think the other reason is that usually a lab has a favorite strain they work on. They are very comfortable in genetically modifying, and for us, it's Jarovia and Saccharomyces right now.
Okay.
One to Petra. Are there questions, or did there arrive some questions online, or everybody satisfied with the information received?
So far, no questions put in the chat, but online participants are now encouraged to post their questions. You can unmute yourself. If there is no questions from the online participants, I have really a very general question to both projects. I think for my place, it was quite obvious that the RRI approach had sort of a beneficial effect to the project and delivered a new input and things to be kept in mind. What is about the other project? I'm posing these questions because I've been writing the ERA CoBioTech proposal, and I've been coordinating this project quite a time. And meanwhile, I stopped, and I see Hannah is also here. Meanwhile, Emily and Hannah coordinated the ERANet, and now it's again Emily and me. So I'm really curious to have some feedback also from from the other project, what the RRI aspect was it of value or was it not of value for you? I mean, it's not necessarily for all projects of value. Just a quick comment.
Sorry, there was this abbreviation, RI, what is it?
Responsible research and innovation.
Responsible research and innovation.
Okay.
I can answer to that. For us, it was really important because the way in which we framed the proposal, to be honest, if we had done the RRI previously, we wouldn't have submitted that the very same application. And it made us realize about, I'm a technology developer, so I'm not expecting to solve a particular problem in one go or make it work at a very large scale. But it felt very clearly after we started the RRI that maybe we were targeting, for example, the wrong type of polymer, because there are very good processes already for doing a very similar thing. And maybe we should have framed the project differently. So that was one thing, is about the technology itself. And the other thing is it also raised awareness about what is the real impact that they could have in people and whether any of those impacts are even known or not. So like the whole discussion we've had about microplastics, it's something that came through having these RRI discussions that we were in our previous.
Yeah, thanks. So I understood that from your project. By the way, I'm wearing plastic today as well, so I will have to rethink that. I'm really surprised that the Sicolim also it was part, I think, of the proposal submission. Yeah, did obviously not benefit from that. That's it. That happens.
Yeah. Thank you very much, Petra, and also, greetings to Hannah. I would say we are really in time, and I think it was an interesting vormittag and morning, and we all deserve some lunch, I think. So for the ones that are now here in the room, lunch will be served outside, and we see each other back in 1 hour. And
no, but I need to interrupt you at that point. I've been organizing in the background via my cell phone something. The other sessions are late because they have more presentations. And I put in the chat for everybody, the next pitch talk session will start at 13:40, not at 13:50. It will start at 13:40 in Berlin 1.
okay.
And for the online participants, I added the WebEx link to make sure that that they have it. So you can, if you want to go back and you don't have it in your papers, you can copy it from the chat. So be there at 1:40 p.m. in Berlin 1.
Okay, and I would say you can just go on with the discussion during dinner, and it's not the wrongest thing to have a bit more time. So enjoy your lunch and see you then.
Thank you.
Thank you, Emily. Thank you. Thank you. Bye. Bye.
Excited to hear science words. It's been a while since I held as we look to singing the talking about synthetic biologists, so exciting to me.
Thank you, Petra.
I work for BBSRC, so